and all this bunch of institutions have been also fundamental for the development of this work. So, right, what I'm gonna talk today is the Elgin reptiles. I'm gonna explain a little bit when and where they live, what are they, why am I bothering to talk to you about this? And then I'm gonna focus a little bit more on one of these reptiles, which is called the Scleromoculus. And I'm gonna tell you about in some more detail about it, because it has an important story, and understanding more about it has important implication on our understanding of the reptile family tree and some iconic groups that we perhaps all know. So the scheme of this talk is that I will start from fossils. You can see, let's see if I can use a laser pointer. Maybe. There you go. You can see a fossil here of what these reptiles look like, this orange blob. And essentially what we have done is taking these blobs, CT scan them, so digitally reconstruct them, isolating individual bones, and then with those new information, we were able to understand more about how the animal live, but also its position in the reptile family tree. So when is this talk taking place? In the Elgin area, which is in the northeast of Scotland, in Moray, there are two separate faunas. One is Permian, late Permian, probably around 260 million years ago. And then there's one which is late Triassic, about 230, 240 million years ago. Why am I underlining this? Simply because between these two faunas, there is a gigantic mass extinction. The Permian-Triassic mass extinction, which is the biggest mass extinction that ever occurred on Earth. And so understanding a bit more of what animal and what faunas were around before and after can tell us about the evolution of life on Earth, but also about the mechanisms of extinction. And ultimately, all these projects related to the Elgin fauna have these questions in mind. So what happens during mass extinction and what we can learn about those. These animals are preserved in sandstone, and unfortunately we do not have an absolute available data, but because of comparison with other similar faunas around the world, we know pretty much when they occur. I talked a lot about mass extinction, but perhaps I should focus a little bit more on why I've done that. So mass extinction have a fundamental role in shaping the ecosystem through Earth history. The most immediate way in which mass extinction do that is by removing species. So we'll see in a little bit that over 90% perhaps of species on Earth during the Permian went extinct at the end of the Permian. That's a lot. However, in the long term, all these vacancies in an ecosystem create opportunities for new groups to evolve, and so to potentially increase ecosystem through time. An example of that is the Permian-Triassic mass extinction, which is what I'm focusing in my current research, actually. That impacted all taxonomic groups, so all possible animals that were around in the Permian were impacted, animals of different size, diet, habitat preferences were affected. And this created a very strong turnover in which up to 96% of the species on marine ecosystem went extinct, and over 70% of land vertebrates went extinct as well. So you can see from the graph down on the bottom left that we have certain type of animals living in the Permian, so synapsid, your Dimetrodon, your mammal reptile-like animal. And then we have a huge depression in the number of species that exist. And then this diversity comes back with Now, Triassic ecosystem are dominated by mainly reptiles. If we look a little bit more closely at what this means what happened during the recovery phase, we have essentially three main groups that occur. Some are the Permian holdovers, lower in diversity. So these are the animals that belong to group that survived the Permian-Triassic mass extinction and continue into the Triassic. We have a Triassic indigenous fauna, so animal that started in Triassic and ended at the end of the Triassic. So animals that are exclusive of this period of time. And then we have what we call the living fauna, and what is the cause of the title of our talk. So living fauna are all those groups that started evolving in the Triassic and survive until today. So things like birds. The origins of birds is to be found into dinosaurs. So dinosaur evolving just after this Permian-Triassic mass extinction. Lizards, turtles, and animals like that are iconic part of modern faunas, all have origin back in the Triassic. And the story about the Elgin reptile fauna is how this animal came to existence. So I promise you the Permian fauna, these look very weird. So you're warned here. On the top here, we have an animal called Dicynodont. It has a beak on the front, two big tusks, and nothing else. These are animals that exist both in the Permian, but also in the Triassic. Pterosaurs are another group, herbivore, massive reptile, parareptiles, so reptiles, but almost reptiles, let's say. And these are animals that also partly survived to the extinction, and we find some relative of these in the Triassic fauna. All of these animals are represented both by skeletons, as you can see from these drawings, but also by a large variety of footprints, as you can see from this lab from the National Museum of Scotland. One of these animals, called Gordonia, has been studied by one of my students, Heidi George, that is now at the University of Bristol. And just to show you the amount of detail that we are able to get from this new technology, this is in blue the brain of this animal, which is over 260 million years old. Heidi was able to reconstruct this shape and compare that with other modern animal. We now have a better idea what these animals were able to do and what their ecology was as well. But I promised you about the Triassic fauna, and that's what I'm going to focus for the rest of the talk. This is mainly my expertise, and I'm particularly attached to this because of their importance to understand the early evolution of modern fauna, as I mentioned before. All these animals that you can see here, from this lizard-like to this giant armored crocodile thing, these are all preserved in the same layers. We know they coexisted together, and I'm very grateful of Gabriel Ugueto for this wonderful reconstruction. So this reconstruction is based on the new data that we have. And actually, if you want to see that by yourself, you can go to the Elgin Museum, and there's a huge print of this available there. Where are we talking about? So this is the world back 238 million years ago in the Triassic. This is where our story comes. So this is what Europe looked like. It was all the continent was mushed together. North America up here, South America, Africa, all attached. And Europe was here with the UK around this area where I'm pointing with a laser pointer. This was an area very different from what it was before. And we know that because of the geology of this area. This is a geological map of the Moray area in the northeast of Scotland. And this is what the deposits where we find the Elgin reptiles look like. You can see here massive sandstone. And these represent desert sand dunes that were advancing. And as they were advancing, they buried the carcasses of different animals that we are now lucky enough to find. Unfortunately, the possibility of finding more of these is quite slim, not because we have exhausted the fossiliferous layer. In fact, my colleagues at the National Museum of Scotland were able to find bone scraps in this layer in one of the quarry. But it's unlikely we'll find more because of the way the sandstone is now quarried. So these massive machines are now scraping the sandstone directly from the walls and immediately reduce them to shards. And these are rocks that are used for gardening around the United Kingdom. So maybe we can find something, but very likely it's going to be reducing very little pieces, which is not particularly helpful. However, we are working with the Elgin Museum to try to make the worker aware of this fossil, and maybe in the future we will be able to do another excavation there. What are the Triassic reptiles? So here we have a simplified reptile family tree. We have a huge number of names. Don't pay too much attention of those. I'm just gonna walk through them very rapidly. This animal is one of those parareptiles I mentioned before, one of the hangover from the Permian-Triassic mass extinction, one of those groups that survived and are still lingering there. Rhynchocephalia, so Brachyrhinodon, is an animal that is its closest relative is now the Tuatara, so in New Zealand. So these are the first lizard-like animals that we see in the fossil record. This is an extinct group of reptiles, Rhyncosauria, and then the rest will all be reptile in the traditional sense that we know. Some of them will belong to the crocodile group, which is called Pseudosuchia. So the first actually four, the Zygnatoides, Ornithosuchus, Herpetosuchus, Stagonolepis, all belong to the crocodile line, while the other two, Saltopus and Scleromochlus, belong more likely to the bird line, so the dinosaur branch of the reptile family tree. That said, why are we talking about these animals? So these animals have been known since the 1800s. So it's a long time. We have a long time to study them. Unfortunately, this is what they look like. This is a fossil here on the left. And as you can see, there's no bone preserved in there. So this fossil preserved in a really peculiar way. The bones are either dissolved during diagenesis, during the formation of the rock, or they are completely powdered. This means that these specimens are impossible or very difficult to prepare, which means that all we are left when we crack the blocks that contain this fossil is these vacuity with these holes. And historically, that's what we use to study this animal. So historically, what it has happened is that some researcher has removed the bone powder that infilled this hole as best that they could. They poured plastic or rubber material on the slab, and then they peeled that off. The results are these peels on the right-hand side. And it goes without saying that to get this sort of information, we have to break the blocks apart, which means that is the destructive technique, which means that we are losing detail. We are losing detail because pieces of sandstone get lost during the process, or when we peel the material from the sandstone, pieces, grains of sands get removed, so we are losing details. But also these plastic material are too dense and too viscous to penetrate into all the cavities. Particularly this animal that we will see later is very small. It's about this size, well, one about 20 centimeter long at best. And so the material that we use to study those is inadequate to understand all the skeleton. Bonus, these casts, these peels are essentially rotting. They are becoming unusable. Many of these are older than 100 years old. Many have several decades on. So this project, a big part of this project was also preservation of information about this animal. So what we did is turn into computed tomography. This is essentially a fancy X-ray machine, but when you go to the hospital and have an X-ray of your hand, that creates a 2D image. Computed tomography, instead of taking a single image, take an image and then the machine rotates, and so they can take another picture, and another, and another, and another. So you come at the end with a series of X-ray pictures that has been taken out the fossil in all directions, and you can use them to reconstruct the what is in the rock in 3D, in 3D. This is great because it's non-destructive, so we don't need to prepare specimens that are difficult or impossible to prepare. We don't need to damage specimen more than what that has happened already, but also we can look at them in more detail. You can look at them on the computer. You do experiment that would be destructive. We can 3D print those, and they are data that are more easily to share than the actual specimen. And also it allows us, as I showed you before, to see structure that are invisible. So your brain cavity or inside the bone structure, this sort of data that otherwise would have not been achievable. A good example of this is this. So this is a fossil you'll see in a minute. You can see some holes here. This is what typically a specimen from the Elgin era looks like. Quarry people in the past were instructed to stop quarrying when they saw something like that and to save the block for study later. So this block has not been prepared, so we are particularly lucky with that we can see inside it. So we CT scan this block, and now you have a cross section of that. This is all that you can see in the surface, continue inside, here in black. And using particular software on the computer, you can essentially color in 3D these cavities and come up with a reconstruction of the skeleton. And that's what it looks like. The first time I saw this, my jaw dropped. So there's an entire skeleton in there that we could not study otherwise, and we are able now to access. You can see the backbones here in blue. You can see the ribs in green, the forelimb in pink and red. And all these purple bits are probably the remains of the last meal of this animal. And bear in mind that this block is no longer than 15 centimeters. So the detail here is exquisite. And this is what the animal looked like. So immediately when I started this project, we took all the unprepared block that we could get our hands on. These are, I think, something like 10 blocks from the BGS. I was able to reattach them together with rubber band. You can see these straight line. These are rubber bands that I used to keep them together. All these block fits together. And once we remove the rocks around, you can see in color all the articulated or almost articulated skeleton of actually two animals. One is similar to the one I showed before. You can see its jaw over here. And the other one is called the Herpetosuchus, ancient crocodile relative that for which now we have much better information. So before this study, Herpetosuchus was known only for the skull and the forelimb, and now we have much more information about the hind limb, the tail, and the backbones, and isolated bones of the skulls. So all these things you can find online, and it's all nice. One of the other animal that I want to show you is Brachyrhinodon. This is one of the CT scans that I showed you, this part of the jaw and the skull. And this highlights very well one of the problems that the previous techniques have incurred into. I highlighted in blue the bones shard of the original bones that were preserved. And I highlighted in red the part that the peels that we used so far were not able to reach. So I'm gonna try to show you what this looks like. So stop sharing this. Now I will share again the screen. Okay, you should be able now to see a orange blob. If you don't, please let me know, otherwise I'm continuing. So you can see different blocks. You can see the crack between different blocks. 1 block, 2 block, 3 blocks. You can see the rubber bands that put this block together. And if I remove the block, you can see a skull. This skull is 2 centimeters long, less than an inch long. And this is what we knew before. So what the peels were able to access. And now in red, I will make appear the new information that we gained with CT data. And as you can see here, it's a lot. So we have a lot of new information, very small as well, that we are now able to study because of these techniques, which I think is pretty cool. So just to orient yourself here, this is the eye, this round thing is the eye. On the bottom, we have the lower jaw, and on the top, we have the skull with the snout on the left. All right. Back to the presentation. Wrong current slide. We should be back at what we are now. So again, this is the skull as with the new information that we have now, with few vertebrae, a lot of information about the lower jaw and new information. But what I really would like to focus today is Scleromoculus. And I'm going to tell you why in a minute. This is arguably the most important of the Elgin reptiles. And that's because it may be key to understand the evolution of pterosaurs. Pterosaurs being this group of marvelous flying creatures, the first vertebrate to achieve powered flight that seems to come out of nowhere. They appear in the Triassic already with wings, they can already fly, and we really do not know very well where they fit in the family tree, which are their closest relatives, which would tell us about their ecology, how they came to be this very successful group. So Scleromoculus, because of the limited information that we had from traditional techniques, have been put in various places in the reptile family tree. It has been considered an animal very much towards the root of the reptile family tree. It has been considered a relative of modern crocodiles, an ancient relative of those. But it also has been considered very close to the first dinosaurs or the first pterosaurs, despite the fact that it really doesn't look like any of them. So if I activate the laser pointer again, this animal on the left, bottom left, is a reconstruction of what Scleromoculus would have looked like based on previous data. And this schematic illustration represents what was the current thought about the evolution of pterosaurs. So Scleromoculus pretty much descending a tree and becoming something that flies. But is that the case? So a lot of what we know about Scleromoculus is difficult to interpret. That's because the animal is very, very small. You can see the hands of my collaborator, Paul Barrett, holding one specimen. And these are actually two skeletons. And I'll show you again. These are, you are seeing well. These are really, really hard to see. And it's been a challenge for researchers to study this animal. However, these things have been described as having a long and flat head, a very short neck, long limbs, a short tail, and a shallow rib cage, which is a combination of characters which is really, really strange for any animal. No animal alive today fit a description like this, which is part of why there has been a lot of confusion about this animal. Yet, some characteristic of the skeleton of this animal tell the researcher that it's really important to understand where it fits in the family tree because it may relate it to the origin of some very important group. So, and altogether, all this uncertainty about the anatomy and all this debate about how this animal look like makes it difficult for us to understand many aspects of its biology. So, did he walk on two limb, on two foot, on the hind limb, or was it quadruped? Was he walking on four limbs? Did he fly? Did he jump? Was he walking around like a lizard? Hard to answer with very limited data. So here's the new data. Here on the left, I'm gonna show you what one of the Scleromoculus block look like. You can see the two blocks smooshed together. The plane where Scleromoculus skeleton is the silver one that I have isolated in the middle animation. This is what the peel look like in 3D, in 3D. However, key difference here is that instead of having to do two different peels, we have now a single one in which we can assess the entirety of a bone. So imagine that a bone of this animal was split between the two blocks. It would have been very difficult to measure it, to understand what one side and versus the other side, whether they belong to the same bone, and all this information would have been lost. With the CT data that we have, I was able to isolate the individual bones. You can see here on the right. And we were able to study not only the bones that we already knew were there in a better way, but we were also able to access to many, many information that were previously inaccessible. Excuse me. So we have 6 specimens of Scleromoculus. This is the digital reconstruction of all of them. I have highlighted in red all the new parts. For example, for this first one, you can see that the tail that curved this way is much longer than we previously thought. We have new information about the foot. And my favorite on this 3914, we have a lot more information. We have an entire arm, part of the skull here, an entire foot, and some ribs. And that's valid for all these different animals. And I couldn't show you the other side of it, but you'll have to trust me that there's a lot more that doesn't meet the eye. So what's new? After understanding a little bit better the anatomy, thanks to the new data, we can now say that Scleromoculus was not such a weird animal after all. We have new data on the skull, which we have reconstructed here like this, like with a beautiful work of Matt Humpage. I'm going to show you a spinning model in just a second. So the skull has been reconstructed in being much deeper than it was previously thought. And that's because we now realize that the specimens were compressed during fossilization. These animals definitely have hands and not wings, so maybe not as closely related to pterosaurs as we thought. The leg and the foot are very long, but also the torso, the ribs are much longer than we previously had data for, which means that the very shallow rib cage that was described in the past, it's something that we have to forget. This animal looks much more like a little dinosaur than we thought before, and you can see that, I think. So let's see if I can convince you a little bit more with this model. Here. Okay, you should be able to see now the 3D model that Matt has reconstructed for me. And you can access this to Sketchfab. Maybe I'll send to Richard the link to this. And you can rotate it. You can see what the eye look like, what these teeth look like. Zoom in. And these are a really, really beautiful piece of work, but it's all based on new data that we have. I enjoy very much playing with this, so I hope you will too. So back to the presentation again. Okay, should be back. What I would like to talk to you. Oh, this is probably my favorite bit. This is a foot with some little claws at the end. And just to give you an idea of the scale of this claw is smaller than 1 millimeter. So the resolution that we get from this model is outstanding, and we see a lot of this information have been used to recreate this model. So about Scleromorphus, so the summary is that this animal is not a direct ancestor of pterosaurs. We understood this by putting all the new information into a data set and then running some phylogenetic analysis with other animals. But it is still a close relative in a group that is called Laguerpetid. So this tells us a lot more about where pterosaurs came from. And I would like to focus a little bit more on the biology of Scleromorphus now to better understand what are the implications of this. Oh, you will see actually the not taking picture and not Twitter. Ignore those. I forgot to remove that. So feel free to put this on your YouTube channel if you want. So with the new information that we have extracted from the city data, now we can answer some more questions about the biology of this animal. And it's something that we do routinely in paleontology. We try to get new data to inform a bit more about the biology and the relationship about the animals. So historically, Scleromorphus has been thought as a hopping animal, like a rabbit, running on two limbs, or hopping like a frog more recently, or again, running with a very short tail, or hopping again. All these interpretations were based on the previous information that we had. So no doubt having short ribs is something that frogs have. And the fact that we couldn't see the totality of the ribs of this animal led some author to think that this was the biology of the animal. However, we were able now to create a better reconstruction, a more complete reconstruction, let's say, where we see that the head is deep, the thigh bone and hip attachment suggests that this animal was not hopping around like a frog. And that's because the ribs are really small. Frog hips are really long and reinforced. So this would have not been possible for this animal. also the thigh bone of this of Scleromoculus attached to the hip vertically while laterally, which means that the leg was coming down vertically, which is not something that is consistent with a lizard-like behavior or a frog-like behavior. So most likely we think that this animal was very good at running and potentially running on two legs. But how do we get to this? We use a couple of measurements, very simple measurements. So I hope you'll follow me here. The first measurement we use is a quadrupedality index. It tells us, it describes essentially the relative length on the forelimb versus the hind limb. And the reasoning behind this is that an animal that walks on 4 legs, its hind limb and forelimb will be equal in size, pretty much. Think of a crocodile, think of an antelope, think of a dog. On the other end of the spectrum, if an animal has a very short arm and very long legs, think about a T-Rex, for example, it's very likely that this animal would be a quadruped, would be a biped, would walk on just his hind limb. So we took all this measurement for Scleromoculus and a variety of other animals that we know what they did. We plot them, and Scleromoculus falls in this very unhelpful interval. So I'm saying unhelpful because animal on the far right, here represented by Coelosaurus, are undoubtedly quadruped. They were walking on their 4 feet. Animal on the left were certainly biped, your theropods, your T-Rexes. Scleromoculus falls in between. So that's a bit of a problem. However, what we can see is that it was probably able to walk at least a little bit on 2 legs, as many animals in this interval do. So because we were not able to remove the dot on this, we looked for another measurements. So which is the ratio between the length of the trunk of the torso and the length of the hind limb. And the rationale between this is also quite simple, I think. So if an animal walks on 4 legs, like this crocodile, this phytosaur here, this ratio is gonna be high because the trunk of this animal will be longer than relative to the hind limb. On the other hand, if you again think of your dinosaurs and theropod, the torso will be shorter and the length will be longer. And this has to do with the position of the center of mass. So a long trunk and a short hind limb will move forwards your center of mass, which means you will be unbalanced forward. And so walking on 4 legs will be more advantageous for you. So again, we plotted Scleromoculus in here, and there's little doubt in this case that Scleromoculus was plots together animals that were walking on their hind limb, so biped. Having understood this, how good of a runner was this animal? And to understand this, we use another ratio, which is the length of the fingers versus essentially the length of the first bone of the finger here on your hand, the palm of your hand, or actually should be on your foot, sorry, and the length of your femur, your thigh bone. So the longest the finger bones, the better these animals are at running. And Scleromoculus, again, there's little doubt. You can see the arrow here, that it clusters together animals that were very good runner. So in summary, I think we can say that Scleromoculus was, let's say, conservatively a facultative biped but certainly a very good runner. And this is a reconstruction that my friend and colleague Gabriel Gato was able to make, and I love the pose of this animal, how interactive they look, and they looking at each other, and the pose, it's really fantastic too. And by the way, all the vegetation that you can see, those are based on real data on these are Triassic plants that really existed at the time. So some take home point: the UK has an amazing fossil record that spans millions of years on Earth, and there are many opportunities everywhere to go and explore it. A good example of this is the Elgin reptiles that lived in two separate periods of times, and they tell us a lot about the evolution of life on Earth. The Triassic of the Elgin reptiles in particular are a really informative window of what the early ecosystem after the mass extinction were, and what the very first member of modern faunas looked like. So even if the ancestor of crocodiles did not look like crocodile as much, that's where the origin of this group are, and that's very important to understand, even for conservation purposes. Finally, I hope I convince you that computed tomography data allows us to work on specimens that we would not be otherwise able to work on. They allows us to provides us information with information that we would not be able to achieve otherwise. And importantly, they provide us with durable and easily shareable data. So even a stupid example like this talk, I would have not been able to share you my 3D models without this spinning data. So thanks to this technology, I was able to take the actual specimen to you that we're looking through a screen. This is just a quick summary of things, and I think I will leave it up for question if you have many. I will leave you with some more of the Elgin reptiles that are in the process of being worked on at the moment. And that said, I'd like to thank you, everybody, and if you have any question, I'd be happy to answer them.
Well, thank you, David, for a fascinating talk. I mean, that's just shone so much on an area.
Glad you enjoyed it.
Well, just to see the detail. I mean, to see that detail in fossils generally is, I think, exceptional. But when they're preserved in sandstone like this, and when you look at the original specimens and the peels and see what you've extracted, it's
Absolutely.
staggering. So
it was a shock for everybody in the team, me included.
I'm sure it was. I guess people will have questions for you. If you want to unmute and ask questions, or you could raise your hand and I'll know if you want to ask a specific question, or feel free.
In the meantime, you think about it, I can show you one 3D print of one of the animal here. So this has been 3D printed. This is a skull of a lizard-like animal. And the actual size of this animal would be probably half of it. So I can increase the detail and have a better look of what the inside looks like as well.
Yeah. So would anybody like to ask the first question? Sylvia, I think you need to unmute.
I've had the pleasure of going to the Elgin Museum to see the reconstructions there. So very impressed. I've also been to the quarry.
Wonderful.
I've got two questions really. One is, how do these wonderful specimens come to be preserved in sandstone, which we always sort of learnt didn't have fossils in? And the second question is it enormously expensive, this computer tomography?
Yes. So let's start from the computer tomography. It is expensive, there's no doubt. It's expensive not only because running the machine is expensive, but also it's expensive in terms of time to reconstruct these things on the computer. And often you can only do that with proprietary software, which have astronomical fees for licenses. However, there are free alternatives to the reconstruction. As far as I know, there are no alternatives for the CT scanning, unfortunately. But there are institutions that have better rates than others, but certainly you will need a bit of money to do this sort of work. The scleromoculus reconstruction of the 6 specimens took me most of the pandemic to do. So we are talking months of work, if not years, which to someone may feel not very rewarding, time rewarding, and but ultimately led to new information, which is what I think was appreciated. In terms of the preservation, I wish there was a better taphonomic study of this animal, because the hypothesis is that what happened is an advancing sand dunes buried the carcasses of this animal. It could be. I'm not sure. Another hypothesis is that this animal actually lived in the desert. I have a little bit more problem with that interpretation, simply because more than half of these animals are herbivore. And in the desert, there's not much to go around, especially in a sand dune desert. So my interpretation here, but I wish I would like to do further studies, that this animal died somewhere not too far from vegetation and water, and then sand dune advanced and covered them.
What you may have, David, is interdune areas with ponded and lacustrine areas.
Absolutely.
Because a lot of the Triassic has rhizoconcretions in many localities, suggesting that plant life was around.
Yeah, it's possible. I don't think there's really evidence for that in the quarries that have been worked on. In fact, these quarries are purely sand dunes from the Triassic onwards. And a little fun fact, this sandstone has been used not only to cover many of the buildings in Scotland. So the National Museum of Scotland is covered by this, but has also been exported for internationally. So part of the new building of the Sagrada Familia has been done with the Permian sandstone from the Clashach quarry.
Can I come back there? Have you linked any of your work with the footprints? Have you managed to get any information, well, from the footprints that are preserved and can be seen in some of the blocks?
Yes, so I've not done that personally. A colleague of mine, Elsa Panciroli, is working on footprints. I hear. I do not know at what stage this is or if it's proceeding, but I certainly hope. Most of the footprints, as far as I know, come from the Permian area, which to me would be even more interesting, because we know way less about the Permian fauna than we know about the Triassic, especially for the small animals. And we have blocks in the quarries of small footprints with drag tails of little lizards, and it would be amazing to understand who made those.
Thanks, Sylvia. There's a question has come up in the chat from Ed King asking, did Scleromoculus evolve after the Permian Triassic mass extinction?
It did, yes. That whole group probably Yeah, that whole group of Lagerpetids or Lagerpetids plus pterosaurs are all Triassic and after. So Scleromoculus is late Triassic in age, but its oldest ancestor, or its oldest relative, is probably 15 million years younger, maybe 10.
Mhm. Yeah. Anybody else have a question for David?
Could I pose a question, please?
Yeah, who's
Lynn?
that?
I'm Lynn.
Lynn? Hello, Lynn. Yes,
Hi.
go ahead, please.
When you're measuring the bones to compare with other bones to develop the ideas of what group they belong to, how do you account for the bones being squashed or broken to get these measurements? Because they're rarely in the normal 3D position that they would have been during the lifetime of the animal.
Yeah, no, absolutely. That's a very good question. So this might take a while, but the main structures that tells us what group an animal belong to can be recognized even if the bone is flattened in most cases. We are lucky for Scleromoculus that we have six different specimens, and so they are yes deformed in a different way, but putting all the information together, if there is a certain structure that is repeated in all these animals, we can be fairly sure that it is not an artifact of preservation. I am more reluctant to comment about ratios. So if I want to understand the exact real shape of a bone, that's way more difficult because again, different type of preservation of this animal. But this should not affect the length. It should only affect how circular the bone was. And some of the specimens of this animal are, the skeletons are flattened, but the bones are 3D, which is another interesting taphonomic event, which leads me to think that perhaps this animal have been exposed on the surface for a while. So animal died, and then it got sort of mummified with the skeleton collapsing but not being flattened. This happened in only some of the species. Some other, the bones are pancake, but others are pretty much 3D. I hope that answered the question.
Thank you.
Audrey, would you like to go ahead?
It seems amazing that there's just this little area near Elgin where these sandstones occur. Presumably, there are Triassic sandstones in other places which might have similar fossils.
I hope so. So in many places that would be buried under hundreds of meters of other rock. The frustrating bit with this for me is that many of the quarries are not exist anymore. So some have been built on. So the old little town of Lossiemouth is being built on the quarries where Scleromoculus came from, which is a shame, but they need houses. So there are other Triassic outcrops in the UK that are promising. So down south, there's the Otter Sandstone, which is pretty promising. The preservation is not the same. Old Red Sandstone is it's more Permian and Devonian, but depending on how it has been subdivided or studied, there may be lenses of Triassic preserved on top of that somewhere. There's some in the inner Hebrides that I really want to go to. And there's quite a lot in the outer Hebrides, well, all the area around Stornoway. That's a deposit of indifferentiated Permian and Triassic. Big boulders that they are coastal, they are very difficult to excavate and looked at, but similar lithology in the United States have bones in there. They're easy to access because they're loose, but maybe if you ever walk up there, you can have a look for me.
Thank you.
David, I think it's in the Trias in Cheshire, there are footprints of, is it Chiratherium preserved?
Oh yes, so absolutely.
Whether that's an area with potential for recovering skeletons of other species, I don't know.
I would say that everywhere you find footprints, someone has made those footprints, so it's worth looking at. On the Isle of Mull, there are a few of those too, and Chiratherium would probably be an animal similar to the Ornithosuchus that I mentioned before. Mm-hmm. So big crocodile-like predators.
Yeah. Okay. There's another question's come up in the chat. Fred Owen has asked, what have you learned from these fossils about the effect of mass extinction on evolution?
Ooh, that's a tough one. Well, we know for sure that ecosystem changed massively. Between the Permian and the Triassic faunas, we have entirely different groups that dominate the ecosystem. In the Triassic, it's mostly all reptiles. So that really kickstarted the evolution of it's a really good window of the evolution of these groups. And then the interval between the Permian and the Triassic deposit of the Elgin fauna, it's a little bit too wide to draw patterns, and we only have 2 data points. So ideally, it would be wonderful to find intermediate stage.
Sorry, somebody has their microphone on. Could they switch it off, please? Thank you. Well, there are no more questions in the chat. Last chance for a final question for David. Susan, go ahead. You need to unmute.
Is that better?
It's much better, much better.
That's a fascinating talk. Thank you so much, David. Going back to how they were preserved, the animals seem to be, the bones of the animals all seem to be quite close together. Mm-hmm. So perhaps not very disarticulated, so they haven't moved far from when they died.
Yeah, I agree.
So does that suggest a rapid burial to preserve them? Or not necessarily?
I think you're right. So the smaller animals are often preserved as skeletons. And to me, there are 2 mechanisms that this could be. So yeah, one is rapid burial. The other one is some sort of mummification. So this is something that occurred to me when I was doing field work in Arizona, and you see a dead lizard has been exposed to the sun for a while. The skeleton is collapsed, but it's pretty much everything in their place and around that. If that thing gets buried suddenly, then something like this preservation would occur. But for sure, very little disturbance have occurred to these skeletons. This is also true for the bigger animals. So we have partal skeletons that are maybe 2-3 meters long. They are less articulated, but they're still mostly there, so which also suggests a rapid burial. So I think your interpretation of a rapid burial is for sure on the spot.
So if it was mummification, what does that mean in this
Well,
context?
I would just think that when you see these carcasses in the desert, the skin still keep everything together, even if it's all completely desiccated. And so in a way, that prevents further disarticulation until the burial comes out.
Right.
So I'm not excluding a rapid burial that is necessary in both cases.
Right. And it implies no predators.
It implies no scavenging.
No scavenging, yeah.
Or limited scavenging, yes.
Thank you. Thank you very much.
Thank you.
Thanks, Susan. Well, I think it just remains for me to say thanks again, David, for an excellent talk. I think everyone's really enjoyed this. It's been quite a different topic for many people, and a real insight into the excellent use of technology to take our understanding forward. So I'd like to thank you on behalf of the Westmoreland Geological Society. And I think if everyone just wants to maybe switch on their screen and their sound, and you could actually applaud your screens and pretend you're in the room. Thank David.
Thank you so much for having me, and maybe next time in person.
Yeah, that would be great. Yeah. All right. Okay. Well, thanks everybody hope you all enjoyed it.
Thank you, Richard.
Thank you.
Great.